Here, what you have is something called a nucleotide. Yes? Nucleotide. We're going to talk about what a nucleotide is. It's made of 3 parts. 1, phosphate, 2, the sugar, and 3, a nitrogenous base. 3 parts to nucleotide. This is phosphate. This is phosphate here. Phosphate is a functional group. It is not this is phosphorus. It's an element. The whole group is a phosphate. The group contains oxygen, phosphorus, and hydrogen. Do you see that? Okay. When you see a drawing like this structural drawing, when you see this drawing and you see these lines that meet, do you notice, is there a letter here anywhere? There is no letter here. Then you know that that is carbon. That means that this is carbon. How do I know it's carbon? Because I'm telling you. If the lines meet, and there's no letter there, it is carbon. How many bonds can carbon make? 4. Look at this. Count. This goes to 1 oxygen, that's 1. This carbon goes to this carbon, that's 2. This carbon to this hydrogen, that's 3. This carbon to this nitrogen, that's 4. 4 bonds, that is carbon. This sugar ends with OSE. Specifically, it's one of 2 things. It's either ribose or it's deoxyribose. Now, to be clear, both of these end with OSE. What are some other sugars that you might have heard? You've heard of glucose. I know you've heard of another one called lactose. So it turns out that anything with OSE means what? Sugar. Anything with the suffix OSE is sugar. So don't be surprised if you see that in the future. This is either ribose or deoxyribose. It is also known as a pentose. In other words, it's a 5-carbon sugar. Let's take a look at what it looks like. A pentagon. Do you see the pentagon? If you count them, that's 1. That's part of this ring. 2, 3, 4, 5. 5 carbons. Glucose is a 6-carbon sugar. It's called a hexose. Lactose, different sugars. All right, so we're good with this sugar business? Now we number the carbons. And the way we number them is we start with the oxygen in a sugar. And we number them clockwise. Clockwise. Follow along. 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. This is going to become extremely important very soon. Literally 1 more page. So it's important that you understand that each of the carbons is numbered 1, 2, 3, 4, and 5. Starting with the O, which is oxygen. I like it that it's oxygen and we start with the O because it looks like 0, doesn't it? So O or 0, then 1, 2, 3, 4, 5. The 1 position, the first carbon in the pentose, gets connected to what's called a base. A base. This is your base. It's called the nitrogenous base. Why is it called the nitrogenous base? Because it has nitrogen in it. Look how much nitrogen is in this thing. It's a lot. All right. So 3 components which we've identified. Phosphate, which remember is phosphorus with oxygen and hydrogen, usually drawn as a circle. Ribose sugar, either RNA or DNA, deoxyribose or ribose. sitting in the middle, and then some base connected to the first carbon. Let's take a look at what we're looking at. These bases can be one of 4 bases, or 5 really. One of 5 bases. There's actually a lot of nitrogenous bases. Let's be clear. There's a lot of nitrogenous bases, but there's 5 that are involved in DNA and RNA for all living things on this planet. That's really amazing. Of all the different kinds of life on this planet, from fungus and bacteria and viruses, all the way up to humans, all of us use the same 4 bases or 5 bases. It's kind of crazy, even though there's many different kinds. This is all in your study guide. A always gets matched up with which one? Does anybody know? A always gets matched up with T, and G gets matched up with C, except for what? That's in DNA. In RNA, A does not go with T, it goes with U instead. So A can go with either T or U depending on whether it's RNA or DNA. G always goes with C. Notice that these are double rings, we call them purines. The G double ring always goes with a single ring. Always goes with double ring, always goes with a single ring. This is called, by the way, this is called base pairing. Base pairing. These are bases, and we're talking about pairs. Are we clear? Yes. These are the base pairing rules. That's all there is to it. Now, how do we know which one's deoxyribose, which one's ribose? Let's be clear on this. Let me speak clearly. You have D means to take away, right? Like detoxify means to take away the toxin. Then you have oxy, which means what? Oxygen. And then you have the word ribose, sugar. 5 carbon sugar. Deoxy means the sugar without the oxygen. Can we all say a duh? So no oxygen. No, it does have oxygen though. Where's the oxygen missing? If this is your O here, where's oxygen missing? Let's see who was paying attention a minute ago. Not the 1 prime. Second prime. I didn't hear you. The 5th prime's up here. It's not drawn. We don't always draw every part. So deoxyribose is missing this oxygen. Ribose has that oxygen. Now let's see who's paying attention. Quick! Is this DNA or RNA? Ribose or deoxyribose? You said RNA. Why'd you say RNA?
Never mind.
Don't say never mind. Be wrong now. Be wrong. Take heart in it and learn your lesson so you do the quiz, you get it right. You do the test, you get it right. Is this DNA or RNA? A lot of you answered already. You're right. I heard you. It is DNA. How do you know it's DNA?
Only a test, only page on the bottom.
There's no oxygen here. All 4 bonds. Look at them. 1, 2. This one's bonded to carbon here. It's bonded to carbon there. It's bonded to hydrogen here. It's bonded to hydrogen here. There's nothing. This carbon is not. Second carbon is not bonded to oxygen. So it's missing an oxygen. It's deoxyribose, DNA. That's one way you're going to tell the difference between DNA and RNA. Here's another way. The RNA molecule is single-stranded. Only 1 strand. Let's be very, very clear. This is a molecule. Molecules are defined as being covalently bonded to one another. The atoms are covalently bonded to one another. Is that clear? So this is a molecule because all of these atoms that are involved here, and you want to see what they look like. It looks like this, right? There are a bunch of these put together. All these atoms that are involved here are sharing electrons with one another. Is that clear? They're all sharing electrons. All the way up and down. That's why we call this a molecule. even though we call this a DNA molecule, really, it's not one molecule, is it? DNA is actually how many? 2. That's why we say double-stranded. We call it one, let's just go with it. But that's because this strand, these are all covalently bonded. This strand is covalently bonded. But what's holding these two together? Hydrogen bonds. These are very weak. Guys, that's really important. Really super important. Because what that means is I can separate these two and not change the sequence. You'll learn about why that's important later. Yeah.
So if you separated them, would each one be RNA?
No. Good question. Why not? What is the ribose sugar here? Is this ribose or deoxyribose? It's deoxyribose. Is this T or U?
T.
So you have T, you have deoxyribose, so it's still DNA. It's just single-stranded. Very good question, though. So the property of DNA is it's double-stranded. It's double-stranded, meaning there's 2 molecules of DNA here. Very important. You'll see why when we start talking about DNA replication, mutations, making proteins. It all becomes very important. And it's being held together by hydrogen bonds. 3 hydrogen bonds between the G and the C. Do you see that? And 2 hydrogen bonds between A and T. That's how we knew. That's how we figured out. That's how Watson and Crick, that's how someone named Chargaff came up with the base pairing rules. We knew that G and C were bonded together and A and T were bonded together before we even knew what DNA looked like. Because there's 3 hydrogen bonds here and only 2 here. We'll talk about that again a little later. So DNA, to be clear, is double-stranded. RNA is single-stranded. DNA has deoxyribose. RNA has ribose for the sugar. DNA has T. RNA has U. 3 big differences between DNA and RNA. You can rewind the video. And let's go on to the next page.